Okay. Thank you very much. The talks that we heard earlier by Peter and Helena make my task a little easier because they touched on some of the key elements that I will be covering on in the talk. This session is about consequences and responses. And what I'm going to be focusing on in this talk are some examples of how we can respond and respond from a scientific perspective to address the needs that we heard about yesterday. Similar to how Peter presented his talk, this will be talking about responding with the world responding in a way that fits in with the world as we find it, not maybe as we would wish it to be. It involves discussing how public sector can interact with private sector to deliver that response. And as the talk progresses and throughout the whole of the talk, we should remember that whatever advances or successes I am talking about are only a very small drop in the ocean of the problems that we heard about yesterday. So the talk will cover three main areas. I'll deal first of all with economic issues, talk a little bit more about some of the diseases that maybe we haven't talked about so far, medical need. I'll go into some detail about the process of drug discovery and development, because I think that's important if you're coming from the scientific or molecular biology background to see how your work can fit into that process. And finally, I'll talk about some of the examples of successes of partnerships and a brief outlook on the future. We've talked mainly over this conference about malaria, TB, HIV. There are many other diseases that have a significant public health impact. These are the diseases covered by TDR, and I'll be talking about some of those or responses to some of those in the talk. There was a question yesterday about TDR's budget, and it was mentioned that our funding matches the disease burden, measured here on this bottom axis as DALYs. We're talking about spending $10–15 million a year on malaria, TB, several $million a year on other diseases. Obviously, this, in one way, it's a significant amount of money. In other ways, it's a very small amount of money. It's a small amount of money even compared to the amount of funds committed to an average university department. But I hope to show you that the impact that that funding can make is significant. I always like to include this component in this graph. This is the disability-adjusted life years, or the disease burden generated due to war and conflict. And if you compare the billions of dollars a day spent on issues associated with war and conflict to the tens of millions of dollars a year that we're talking about here for neglected diseases, maybe there's some imbalance in our priorities somewhere. The reason why TDR, or public sector, is involved in product R&D at all is illustrated by this graph. This is a distribution of the pharmaceutical market by value. For high-income countries, that's US, Europe, Japan. Middle-income countries, Brazil, India. Low-income countries. And you can see that the huge volume, the huge size of that market is in the north. And any company wanting to invest in this market will go to the diseases of concern of those countries. A lot is made about the markets in India, Brazil, the emerging economies. Even there, we're talking about a very low proportion of the market value residing in those countries. And it is just as difficult a job to get pharmaceutical companies in middle-income countries to invest in diseases of the poor as it is to get companies from the north to invest in diseases of the poor, because the same commercial realities that drive the company are the same. Our problem is that most of the diseases we're talking about only reside in the low-income countries, where there's extremely limited incentive, but where the need is very great. So the mission of TDR is essentially twofold. It's to undertake R&D for new tools, and at the same time, as Yaya Toure was stressing yesterday, to strengthen research capabilities to where these diseases are endemic, so that the countries and the scientists in the countries themselves can begin to take some degree of control and ownership of the process. If we're talking about product R&D in any sense with respect to this activity, it is important to realize that it's sandwiched between the basic research, the type of research that's carried out here at the EMBL labs, and implementation research, taking the tools and trying to make them work, the sort of work that Paul Farmer was talking about yesterday. And again, from a philosophical point of view, from a practical point of view, from a self-enlightenment perspective, to have all that based on a background of capability strengthening within the countries where we're wishing to operate. Now, within TDR, this product research and development arm, which sits between basic and implementation research, is itself composed of numerous projects, and each of those projects are little mini public-private partnerships. Most of them involve interactions with academics and industry to drive certain development projects forward. There's a growing interest and development of work in the diagnostics area, particularly TB diagnostics, and there is some activity also in the vaccines area. However, for this talk, because of the time and also because we have more funds and more experience of developing drugs, I'll focus on drug development for the purposes of this talk. The first thing that you need to be aware of as you set out to develop a product, any product, is to define its profile, to know it is what sort of product you want. Now that sounds a very sensible thing, but a lot of people, when they start out in the lab or to research something, or they think they find something, do not have any idea of what the end result or product is that they need. For malaria, we all know about the high disease burden. We're talking about a disease that is primarily for people at risk, young children under 5, pregnant women, travellers, migrants. This need to treat young children under 5 from a drug development perspective raises particular issues of safety. It's also important to know what you have to treat a disease, because what you have to do, as Peter was saying earlier, is to come up with something better. The problem with anti-malarial drugs largely is due to resistance, but it's not just resistance. There are safety issues, there are compliance issues. Compliance often results from a short half-life drug that needs to be given over many days rather than a few days. So in trying to design your drug, you're after efficacy, but you're also after appropriate half-life pharmacokinetics so that the drug can survive in the body for long enough, and particularly important is safety. And safety has a particular importance also when we're talking about treatment in the environments that we're talking about, particularly in Africa, where essentially non-prescription use is the norm rather than the practice. So if you take what Peter Goodfellow was talking about just now with respect to inappropriate use of antibiotics in North America, Europe, transfer that to what's happening in resource-poor situations in Africa and Asia, the problem is manifested many-fold. So for antimalarials, we're after drugs which are orally bioavailable, which you can treat within 3 days, safe for use in young children. There's a desperate need for more drugs that are safe for use in pregnancy. We need improved treatments for severe malaria. There's a type of malaria called Vivax malaria, which predominates in India and certain parts of Asia and Africa. I won't go into that in too much detail, but there are further complications. You're not often just treating one species of malaria. You need drugs to treat many species. And although at the moment there is sufficient medicines around for prophylactic use, maybe in 10 years' time that might not be the case. And so for travellers and migrants, there might be a need for improved prophylaxis. Another disease I'll talk about is leishmaniasis. This has many manifestations. It's a parasite that you get infected from a sandfly bite. If it infects macrophages, if it's on the skin, it gives rise to this sorts of lesions. Mucocutaneous lesions of this type can develop, and if it's internalized, it can lead to a wasting away and death. The drugs that we're using to deal with these are over 50, 60 years old. We're using drugs from the pre-antibiotic era. All of them have problems, all of them with safety, all of them are injectable. So you're talking about having to treat a disease by injection over a 2-week period in a resource-poor setting. Urgent need for oral drugs. Carlos mentioned miltefosine, I'll come on to that later, and urgent need for new types of drug with formulations that allow treatment of cutaneous and subcutaneous leishmaniasis. African trypanosomiasis is a growing problem in Central Africa. Parasite, bite of the tsetse fly, gets into the bloodstream. If it crosses the blood-brain barrier, then it can lead to coma and death. This lady here died shortly after this photograph was taken. She did not actually die from the disease, although she had the disease. She died from the toxicity of the drug that was used to treat her. The main treatment for African trypanosomiasis as we enter the 21st century is an arsenical. It's the only arsenic-containing drug that I'm aware of that is allowed for use for any disease anywhere. It's this one, Melarsoprol. Okay? Again, pre-antibiotic era. 1920 drugs, Suramin still used, 50s. The one that I've put the ring around there is an injectable, and the reason I put a ring around it, it's 1990, and that was a drug in which TDR was involved in the development of. But a huge unmet medical need there, a need for drugs orally available for early stage disease and injectable for late stage disease. Another sort of disease that we deal with, filariasis, helminthic infections. Helminth infections of the lymph can give rise to these. Pathological response. Onchocerciasis, river blindness. The adult worms reside in nodules in the skin. The microfilariae can migrate to the eye, and the pathological response there leads to blindness. Again, with the exception of ivermectin, which was also developed through a TDR collaboration, we're talking about drugs from the pre-antibiotic era. And there's a desperate need for drugs that kill the large adult worms as well as the small microfilariae. And we're working, as was mentioned, with albendazole and ivermectin together in combination. So as we look at these issues, there's two key problems. When we're dealing with issues of access, we need new money, new thinking, partnership campaigns, involving also local communities in that process so that the people that are being treated have an ownership. Again, the type of issues that Yaya Toure was referring to yesterday. For R&D also, we're talking about needing funds and new thinking, and particularly how we can interact with the industry. Now, before you can start to interact with the industry, you have to be aware of the process of drug R&D, to be aware that it is a highly risky business. We start with early blue sky research here. That has to be rapidly filtered down to individual molecules that can be tested in humans and ultimately result in a drug. As you go down this chain, the value of what you have for health is added and your risk is decreased. But the plethora of opportunities here that you can engage in are all incredibly high risk. As we also heard, you're talking about a 10 to 20 year process at a cost of hundreds of millions of dollars. Most of that cost of hundreds of millions of dollars is due to the failure of the projects in which you're engaged. Now, an individual finance officer of a company is looking at this. He sees it in terms of the red line being the cost over 10 to 20 years, and subsequent to that drug being available, he has to get a return on that, preferably before patent expiry, to cover that cost and earn interest over that cost. This equation doesn't add up for the diseases we're talking about. And the deficit is actually in the millions and millions of dollars. There's two ways in which the public sector can engage in trying to address this. One is called a pull mechanism, trying to increase the revenue or guarantee purchase or pay higher prices for product at this end. And the other is the R&D effort, which is to try and reduce the cost and risk of R&D. As we're an R&D organization, we focus on this push mechanism. And essentially what we're trying to do with our processes are to lower the cost and risk of a company engaging in this R&D activity. In return, we have agreements through which they guarantee affordable pricing and they accept a lower return at this side. All of the partnerships that I'm going to discuss are based on memorandum of understanding and agreements where this basic pact is the basis of the understanding. For the process of turning biology into drugs, we start at this early stage, genomics. We know that if we get the total genome, we have the basis of all the information that we need to understand the parasite's biology. We go from genome to transcriptome to proteome to metabolome. Metabolomics and an understanding of the comparative biochemistry of the parasite with the human host is the key to good, effective drug discovery. And the tools that allow genetic validation, knockout, and also linking to chemistry so you find compounds that can selectively inhibit certain targets to give validation are the key to defining what molecular targets you can move forward with for drug discovery. For malaria, we're now in the lucky situation where we have the genome, we have a huge amount of information of the parasite biology, we have a great deal of knowledge on targets that exist in novel organelles like this apicoplast, which is derived from a green algae and has its own genome, a lysosomal food vacuole where hemoglobin degradation occurs. There's a rather odd mitochondrion in the malaria parasite that has different properties to most mitochondria, and there are various other aspects there. So there is no shortage of targets to better understand and to better work on. For example, the apicoplast genome, this organelle derived from a green algae many eons ago, was found when it was sequenced to have prokaryotic transcription translation apparatus. That explains why some antibiotics inhibit malaria growth. Now, if we can optimize some of these antibiotics for malaria use, we might be able to get very high quality anti-malarial drugs. We have leads to work on. Carlos mentioned yesterday about fosmidamycin. In humans, we make isopentenyl pyrophosphate through this mevalonate pathway. That pathway does not exist in the malaria parasite. Instead, they have this DOX-P synthase pathway, which exists in bacteria and plants. And lo and behold, Fujisawa had already had a compound, fosmidamycin, which inhibited this and has been found even in the clinic to have some level of antimalarial activity. Whether it is good enough to be a drug or not is not clear. But this is the power of genomics giving you a quick lead in to a potential new drug. Hemoglobin degradation. This is important because chloroquine, mefloquine, quinine, artemisinin, all the best known antimalarial drugs are known to inhibit this pathway. So by finding out more about the molecular biology of this pathway, you identify more targets that you're fairly confident can lead to a drug. So that's for malaria. Similar curves, graphs, slides could be presented for TB. It's also important to notice that all of these parasites that I've discussed have ongoing genomic programs associated with them. Many stimulated way back by TDR that have now been taken over by other funding agencies. So within a very short space of time, we are going to be in a situation where we have the genomic information for most of these parasites. But that's just the first part of the story. The next part of the story is where it starts to get difficult and where it starts to move beyond the realm of the molecular biologist alone. And also beyond often the experience and the knowledge of molecular biologists to know how to take things forward. Because this is where we start to talk about discovering compounds, and it's where biologists have to start interacting with chemists. You can't get a drug without chemistry. And we were talking about functional interdisciplinary interaction. The most important interdisciplinary interaction that can occur for drug discovery and development is the interaction between academic biologists and academic chemists for these diseases. People sometimes think of drug discovery as a linear thing. You have a target, you get inhibitors, you screen, you screen, you screen, and you get a drug. It's more complex than that. Once you have your target and a lead, often there's 3 to 5 years of focused medicinal chemistry involving maybe 10 or a dozen chemists working full-time, producing hundreds of compounds before you get a compound that you can take to development. And then it's a huge amount of time and risk and money before you actually get a tablet. Again, the central importance of chemistry is critical. TDR has over the years developed a number of parasite tests for screening. We organize high throughput screens with some companies, and we can do some limited PK analysis. But to really make drug discovery work, it's important to have multidisciplinary projects and teams working on these projects. And that led to an experiment which was started in 1998 out of TDR called the Medicines for Malaria Venture, which had multiple partners, involved industry in the early discussion, and has recently spun off as an independent not-for-profit foundation in Geneva. The goal here is 1 new drug every 5 years. We're talking about a budget of 30 million a year. You're talking about 20 projects. This 30 million is not the full cost. This is part of the cost. Other parts of the cost are offset by in-kind contributions from both academic and industrial partners. And in the space of 3 years, you've got about 11 or 12 projects running, which starts to look like a portfolio that, even with the risky nature of the business, could deliver 1 new drug every 5 years. That portfolio probably needs to double in size before it can do that. And you're talking, within those projects, maybe 20 full-time scientists at around $1 million a year to make that happen. I just want to briefly, what that has allowed to happen is that the potential target coverage for malaria is now starting to be covered by these projects. So there's still opportunities for new targets, but several of the promising targets are being covered by drug discovery projects. I'll just give you a few examples of how these work in practice. This is one involving GlaxoSmithKline, London School of Hygiene, Bristol University, UK. You've got a team of biochemists working at Bristol that have an enzyme structure, biochemical assay development. You've got a team of chemists working at GlaxoSmithKline in Madrid, doing high throughput screening, getting leads, manipulating that chemistry, passing the compounds to Bristol to screen. And we have at London School parasite assays, testing against the parasite in Petri dishes and also in mice. Collectively, that forms a drug discovery project which is working quite well and quite effectively. And I think these projects can be as high caliber as anything that exists actually within the industry. Another project which I'll spend a little bit more time on is a more downstream project where there was a lead that was wanting to be improved. This graph here is a graph of what happens if you infect a mouse with malaria. The black line is the mouse gets a high number of parasites. If you treat with an artemisinin derivative, you get a rapid cure and a recrudescence after about 2 to 3 days. Artemisinins in the clinic, you need to treat for 5 to 7 days, which is too long. So the goal of this project is to get something that is as good as an artemisinin in terms of efficacy, but has a longer lasting activity. And this project is starting to achieve that. Again, linking up chemists at Nebraska, screening at Swiss Tropical Institute, pharmacokinetics at Monash, toxicology and project management from Hoffmann-La Roche. The reason why this project is quite important is that it recognized early on that the major reason most projects fail in the clinic is not due to efficacy, it's not due to toxicity, it's due to the pharmacokinetics of the compound. The compound doesn't get absorbed, doesn't get to where it is needed, doesn't stay in the body for long enough to do the job. And these are called ADME properties: absorption, distribution, metabolism, excretion. So what you have to do with a drug is to get a compound that inhibits your target, but also is absorbed from the gut, is not over-metabolized by the liver, and stays in the blood for long enough to do the job. And after it's done its job, is excreted so that it doesn't hang around in the body to cause toxic side effects. There have been huge advances in the chemistry that allow that sort of manipulation to happen over the last several years. And what this project did is it took a compound that had highly effective efficacy and started to manipulate the other part of the molecule to deliver on the pharmacokinetics. So again, this is our mice that's infected. This is what happens with artesunate. This is what happens with a three-day treatment chloroquine, a 2-day treatment mefloquine. By manipulating the chemistry, well, this is what they started with, something rather like artesunate. By manipulating the chemistry, you have a compound now that is roughly on a par with chloroquine. They got some compounds that were even longer lasting, more like mefloquine, and they even got one compound that with a single dose cured the animal completely. This product might not be useful in every malaria situation, but you imagine if you're in an emergency situation in a refugee camp and you can give somebody one tablet, they take it and you know that they've been cured of malaria. It has terrific public health advantages, and that's the sort of thing that this molecule might be able to do. So essentially what all this work has done is it's taken a situation, say with the artemisinin, where you have the product available over the minimal inhibitory concentration line for a short period of time, to a situation where it is over that inhibitory concentration for a longer amount of time, both because you've increased the oral bioavailability of the compound, you've increased the half-life, and also in the process you've increased the potency of the compound. Engagement of chemistry and the biology with this chemistry, both through targeting of activity against enzymes and in vitro assays, and with pharmacokinetic and animal assays, is as intellectually stimulating and rewarding as any other academic process that I know of. You can turn this into an academically rewarding process as well as yield a public health result. Even if you have such a compound, you're still at risk of failing. This is just an example of a compound I was involved with when I was at Roche a number of years ago. We must have sunk about easily $10 million into this compound. It was a superb compound, had superb efficacy, was able to treat in very low doses, but turned out to be phototoxic, and we had to stop the project. So you can go quite a way along down the line, and a result like that can wipe out tens of millions of dollars of investment. Next stage is development. People think of development as being clinical phase 1, clinical phase 2, clinical phase 3. It's a bit more complicated than that. You have to carry out a lot of preclinical studies. Those preclinical studies don't stop when you enter the clinic. And in addition to doing preclinical studies in animals, you're also talking about doing chemistry pharmacy work that also has to feed in. So it's quite a complex business to take a molecule from here through to here for registration. And when you've got to registration, you've still got the huge issues of implementation that we heard about yesterday. Again, the way this is done is through having virtual teams. This is just a list of the sorts of people that are present in TDR. You know, people expert in preclinical, clinical development, planning, regulatory. Critically important is that most of the work is done by outsiders and partners, both personal and institutional, that make this happen. This is to coordinate, this is to do. To talk about some past successes, just to show that this model can work. People often say, we need to do things, and there's a promise of doing things. This model has been demonstrated to work. A very small number of compounds have been developed over the last 25 years for tropical diseases, but we have been involved, TDR has been involved in several of those. I've put a square around the ones that I think we can say have had a strong public health impact. Not every drug that's developed by a pharmaceutical company makes a large sale. Not every drug developed by this process will necessarily have a high public health impact, because you don't know how good a drug's going to be until you start to use it in the real world. But the three I've chosen are eflornithine, ivermectin, and this compound, miltefosine. Ivermectin, which was mentioned earlier, it makes its money from treating pet dogs that have dog heartworm. And there's far bigger market for Merck in dog heartworm than there is for onchocerciasis. The net result of this was that upon discovering that the compound was active in onchocerciasis, upon assisting Merck with the studies, Merck have decided to donate this compound for onchocerciasis control. That has led to a partnership involving many countries, involving many foundations, based on this one discovery. And since this drug was introduced, the value or the incidence of onchocerciasis has dramatically increased. New farmland that was previously unusable is now, next to rivers, is now usable because of this drug. The drugs also, has been mentioned by Peter, being used also in combination with albendazole from GlaxoSmithKline to try and address the issue of lymphatic filariasis. And there is now a lymphatic filariasis eradication program based on the application of these two drugs. So, some basic science, some appropriate testing, and you have a drug that can make a huge public health impact. The other drug that's had a huge impact is eflornithine for African trypanosomiasis. This is called locally often the resurrection drug because people are near death. When they receive an infusion, they come back to life. That's a partnership with Aventis and with MSF to ensure continued drug supply. And finally, the drug that Carlos mentioned, miltefosine, which is the first ever oral drug for the treatment of visceral leishmaniasis. And we're not at a stage yet where we can say that this drug has had a huge impact, but the fact that it can be applied orally means that the Indian government are very interested in seeing if it can be introduced into programs. As with any drug, there are issues. The major issue with this drug is contraindication in pregnancy. So there is a fetal toxicity associated with the drug, so we have to have programs set up and established that can filter out pregnant women and treat them with a different drug. So there are now, very rapidly after its registration in India, detailed phase 4 studies set up, cross-border Bangladesh, India, Nepal, to see whether this drug can work as well in real life as it did in the clinical studies. And if they prove effective, then this drug could also start to have a huge public health impact. There are other projects going on in this area. This is TDRs. I showed you the MMV portfolio. There's also a portfolio being developed by the Global Alliance for TB Drug Development for tuberculosis. And by working with partners, both big pharma like GSK, small pharmaceutical companies like Shinpun in Korea, and we're also talking now with several Indian pharmaceutical companies, getting them involved. You can start to gradually introduce new products that can make an impact. Some will be new products like this Lapdap, a new antimalarial, which we hope to introduce next year. Some are taking existing products and doing studies that enable them to be used on different patient populations, particularly young children. And some, again, future are combining drugs in new formulations. In all of this work, it is important that, particularly with the studies that are undertaken in developing countries, that great care is taken to build the capacity while you're there. That involves following good laboratory practice for all preclinical studies, and particularly good clinical practice. The work that we've done with miltefosine has led to the training of large numbers of clinical practitioners that are now being requested by other agencies, other bodies, and even pharmaceutical companies to carry out work for them in those countries because of the training that they received during the course of this development. And ethics was mentioned yesterday. In working through with GCP, we've found that one of the critical factors limiting the ability of institutions and countries to control or have an appropriate say over the research that is conducted in their countries is their ability to ethically review the work from their perspective, not to have to rely necessarily solely on the ethical review of bodies outside their country. And there's an initiative recently started called the Strategic Initiative for the Development of Capacity for Ethical Review. This is a bottom-up approach to ethical review. Regional fora are being established in Asia, Africa, South America to discuss these issues and come up with their own solutions. And there's been a growing interest in that even in the States and in Europe in this model of improving ethical review. To finish, 2 more slides. I think the first is to say that putting money focused into product research and development, this link between basic research and control, if you look at how much it costs, it sounds like a lot of money. But if you consider how much money is invested in basic research and how much money is invested in implementation of the existing tools that we have, often ineffective tools, then it's money well spent. For tens of millions of dollars a year budget, you can have a huge public health impact. What it requires is both government and international commitment, industry commitment, and funds. It also requires an appropriate management structure to allow this to happen, because we're talking about linking and partnering with people from different organizations, different cultures. And in addition to TDR, as I mentioned, there's MMB, JTB. The Microbicides Initiative has recently been launched. There's an AIDS vaccines initiative. There's a malaria vaccines initiative. There's a new MSF-led Drugs for Neglected Diseases initiative. All of these are hopefully going to have an impact on public health. There is, however, I think going to be some need to keep all of these activities somehow coordinated and linked in such a way that they do deliver for public health. There needs to be some validity associated with these organizations that they are acting on the basis of public health. But there's a great expansion of activity in this area, and I think it augurs well for the future if the funds can be maintained. That's at the macro level. At the micro level, and this is coming back to Helena's point, it requires, for all of this to happen, for scientists at the early stage to be engaged and to be aware of the processes that follow on downstream. The reason why there is the excitement to do drug discovery is because of all the work on the genome and all the work that has come out of the genome. It may only be that 5% of that work has a potential application, but that activity has generated a lot of excitement that the downstream can feed off. For neglected diseases, it also is going to require scientists at the academic level to become more involved in the downstream work. For Alzheimer's disease, you give a target, pharmaceutical company will take it away, say, thank you very much, we'll work on that. For this, if you're to get pharmaceutical company engagement, public sector scientists are going to have to stay with the companies all the way down the line. Companies do not have the expertise to develop a leishmania drug. They don't know what leishmania is. The public sector have the clinical expertise and know what leishmania is. And finally, as was mentioned yesterday, science funding agencies also have to kick in and be prepared to fund this downstream activity. All the organizations I put on the previous slide, all of them receive funding from foundations like Gates or Rockefeller, and development agencies like GTZ, Swiss Development Corporation, USAID, DFID. The basic science funding agencies tend to stop short of moving that extra leg, and they tend to stop short, with the possible exception of the NIH, of partnering other organizations. So there requires a cultural shift in the way science funding agencies view their responsibilities in taking this work forward. And at the end of the day, to come back to the very first talk yesterday that Paul Farmer made, all of this has to be done not just with the thinking of terms of the technical properties of the drug that you have, but the fact that those technical properties are meeting a public health need and, more importantly, meeting a patient need. And if you are meeting patient needs, then the public health needs tend to take care of themselves. Thank you.
Thanks very much, Rob, for very specifically addressing what can be done and how it is currently being done in some successful examples. We're a bit over time, but I think we have time for some questions.
Thank you. This slide that you showed with the market segments of the low and the middle income countries, it seemed to me to be a sort of a pessimistic, if not a sort of a short-sighted view, because it neglects the market potential. If you include countries among the low and middle income countries, countries like China and India, then you have billions of people that could use these drugs potentially. Some people might say in some cases the diseases are endemic and the people don't develop symptoms. But this was the case in Egypt, for example, I think was amoebic dysentery until the 1980s. And then sometime around the middle of the 1980s, the Egyptians started developing the symptoms. So these things can change too.
I think they can change, and people's perspectives can change. What I was showing you is the current situation. And I think what I was telling you isn't necessarily what I would like to be the case, but it is the standard response of people investing funds in pharmaceutical research.
Yes, you mentioned this new mechanism being developed for ethics evaluation. Yeah. Could you tell us a little bit more about how that will be organized and put into effect? Okay. That is to say, who will be the people who will actually assess the ethical perceptions of people in different places, and how will they go about doing it?
I wasn't talking about setting in place a new system for ethical evaluation. What I was talking about was encouraging the development in countries and institutions where there is now a limited capacity for them to review, to institute ethical review, to start to develop that capacity to institute ethical review. So it's not any way an imposition or a dictation of what the rules and norms should be. There have been documents produced that outline current best thinking and practice, but solely in a guideline perspective. It would be for the organizations, nations, regions themselves to debate and decide how best to implement their ethical review. It's not an institution. It's a network. It's a network.
TRIPS, the international treaty on intellectual property pushed forward by the World Trade Organization, has generated quite some concern, especially for low-income, middle-income countries. How do your partners see the implementation of strong, or at least some intellectual property rights in the countries where the market is? Is it important for them, or to put it the other way around, can they compete with generic producers if they want to market their drugs?
The projects that I outlined, all of the products were covered by patent for those companies involved. In the malaria GATB projects, the discovery, there are obviously intellectual property rights involved, and so all of this work is done in full accordance with standard intellectual property practice. I think however, it will vary from product to product how much of that intellectual property ultimately is retained. Probably you'll find that companies will take an effort to maintain their patents in European countries where there might be a prophylactic return, possibly in some countries of production like Brazil, India. They will probably not bother too much about maintaining patents in other countries. Most of the companies that are engaged, certainly the big companies, do not engage in this really in any way, shape or form to gain a sort of a profit back. They will probably make a loss. The small companies do need to make a bit of a profit, and certainly to more than cover their costs. But everything that I've done and said is fully compliant with TRIPS, both with the mainstream and ultimately the possible exemptions that exist.
One more question.
Now, you pointed out the chance of genomics and other omics approaches to research in infectious disease. One of the beauties, it's quite cheap. All you need is computer, internet access, and some smart guy sitting in front of the computer. And I would say if it came to someone sitting in Africa doing this research compared to someone sitting in the United States and Europe doing this research on infectious diseases in tropical countries, I would put my bets on the guy in Africa. But you still have the problem that you have these bright students often wandering away. You've still got the brain drain problem from the developing countries to the developed countries simply because there they get the funding. Wouldn't this also be a task or a challenge to overcome this and establish a research base in the developing countries?
Yeah, I mean, I think that's related to some of the discussion that occurred yesterday. One of the things that the strategic research unit in TDR is focusing on are networks of bioinformatics networks that can, and putting a lot of effort into training into bioinformatic technologies so that scientists in developing countries can utilize the genomic information. As you said, I think, and there is also training for scientists to leave their countries, study, and go back to their countries of study as part of research capability strengthening. As you said, the main thing is to try and this is something bigger than anything that one organization can do, but to create an environment where they can return and have a satisfying career. I think in India you're starting to see that happen now, but for many of the countries that we're really concerned with, we're a long way from that.
Okay, I think we should continue now.